So what I'm going to be telling you about today are regulatory elements in the SARS-CoV-2 genome that are embedded in the RNA of the virus and not proteins. And so I think a goal I have for today is to have you shift your focus away from what we usually think about, which are the proteins of the virus, to the important elements that we're discovering that regulate viral function that are in the RNA. And so the question is, how do we find these structures in coronaviruses? What do they look like and what do they do? And this is just, this picture I'm showing is just a chunk of the hepatitis C virus to show how we think about RNA. It's a little different than most people. Let me just a second. Sorry, I'm having a little trouble with my screen. I'm going to stop sharing for a moment. Sorry. Okay. Let me make sure I can still move. Yeah. Is everything okay, Andy? I'm going to assume.
That's perfect.
Okay, good. So the prevailing view that most people have of the way viral RNAs and viral RNA genomes look like, even positive strand RNA viruses, is this plate of spaghetti, and especially in open reading frames. That's how we think about it. And I'm going to try to convince you that it looks more like what we see on the right, that there are modules of structure that have discrete secondary and tertiary structure, and that this matters for function. Now, the first hint that this was true came from work from the Simmons lab, where they did something called a calculation called genome scale ordered RNA analysis, or GORS analysis. And what this was using sort of at that time reasonably crude RNA secondary structure prediction algorithms genome-wide to figure out how much propensity for base pairing there was over random chance. And they calculated something called the minimum free energy difference for a given stretch of RNA for its ability to form base pairs relative to random sequence. And if you have a z-score of 0, that means there's almost no structure relative to random chance. He also saw that in large DNA viruses, base pairing didn't really deviate much from random chance. And flaviviruses didn't appear to have much structure content. But the field got a big shock when he did this calculation on hepatitis C virus, which, as you can see, has a z-score around -3, which indicated at that time it was one of the most structured RNA genomes ever studied. So the next thing the Simmons lab asked was very important. It was not just, is there structure, but is the structure important? And are these discrete structures important? And the way he did that is quite elegant. What they did is they hypothesized that this minimum free energy difference over time as sequences diverge should just go from high levels to 0. In other words, if it doesn't matter what the sequence is, it should be possible for these secondary structure elements to just sort of decay with time. But as you can see, for different strains of Hep C, this is not true. They maintain a high level of minimum free energy or stable base pairing content, which is what this means if this number is high, even as their sequences diverge, which means that even as they diverge, they're maintaining discrete secondary structures. So this was very exciting because it meant that positive strand RNA viral genomes had hard-baked structures within them that were essential. So before we talk about SARS-2, I'm going to tell you a little bit about Hep C, because that's where our understanding of how to think about these issues really began. I'm going to show you the whole sequence of Hep C just as a cartoon. It has 5' untranslated regions and 3' untranslated region. And in the 5' UTR is an internal ribosome entry site that's necessary for translation of a large open reading frame. Now, you've got to realize Hep C is what we used to think was a giant RNA. It's about 10,000 bases long. As you'll soon see, it seems kind of tiny now by comparison, but at the time it seemed really big only a few years ago. And we knew there were important structures in both of the UTRs, and in work by Ralph Bartenschlager and Charlie Rice, and in my lab, we knew there were very important secondary structures in the open reading frame itself. And there was a code within the code for function. And this ORF here is made into a giant polyprotein that is then cut into all of these different structural and non-structural proteins. But Hep C is pretty simple because the positive strand genome is just one big RNA. But until recently, we actually didn't know the discrete secondary structures in that. But I'll digress a little bit to tell you a little bit about an important contribution that Charlie made in all of this. And that is that without an understanding of the RNA secondary structure in the 3' UTR, we would not have the HCV drugs that we have today. And one of the important things Charlie succeeded in doing was that when he initially began trying to come up with a clone that would replicate in cell culture for Hep C, he kept failing, and he wondered if perhaps there was a mistake in the sequence and predicted structures in the 3' UTR. So the lab rolled up their sleeves and recharacterized the 3' UTR and found indeed known sequences were incorrect. So they fixed it, and then they were able to come up with a strain that was able to replicate in cell culture. And because they actually focused on the RNA, they were able to optimize a cell culture system. Previous to this, you could only study Hep C in infected chimpanzees, so that wasn't very good for drug development. But with a cell culture system, it was possible to do the screening and development and come up with the drugs that now cure Hep C. So what I'm showing you now is some unpublished data from my lab where we have determined the entire secondary structure of Hep C in infected cells. So this is the secondary structure in vivo. And you can see the regions of base pairing and single-strandedness. And we've published about half of this in 2016, but we've been able to complete much more of it in vivo, and that'll be published soon. And you can see that it's incredibly complex, even in these open reading frame regions. I'm just circling here some pseudoknots that we identified recently and many others, and we've proven that they're important by making compensatory synonymous mutations that don't disrupt translation, but they do disrupt the secondary structures. And all of these, many of these secondary structures have specific roles in replication, packaging, infectivity. So this was where we got started. And then, of course, in the spring, a bigger problem landed on all of our desks. And this was intriguing to us because the SARS-CoV-2 genome is 3 times bigger than Hep C. It's coronaviral genomes are about 30,000 nucleotides in length. And instead of one nice, simple RNA to work with, there are 10 of them, maybe probably many more than 10. And so we thought we should start attacking this problem. So we pivoted and began working on this immediately. And let me just give you a very brief introduction. You probably already know this. The genomic RNA, this 30,000 thing, is made up of ORF1AB, which encodes the replicase genes. And then through a template switching event, a whole additional family of RNAs is made that make these subgenomic RNAs that encode many of the structural proteins that are important for the virus and that are cartooned here. And in the interest of time, I'm not going to tell you about what they all do. I am going to come back and tell you a little bit about the N gene in a moment. But you can see that the problem has now been amplified in terms of understanding RNA structure. It's a big giant RNA, and it's not just alone. It has many other partners. So to begin attacking this problem, we decided to repeat something very similar to Simmons-Gorse analysis. And we obtained results that were very similar in terms of the base pair content of SARS-CoV-2, just the genomic RNA. We wanted to do that calculation, but first we wanted to repeat what Simmons had done on flaviviral, human, and HCV RNAs, and we got very similar results, although we calculate the Z-score a little differently. You can see Hep C as predicted, is very different and much more structured than you'd expect from random chance. But now let's look at SARS-CoV-2. It's 50% more structured than Hep C, which previously was thought to be the most structured genome on Earth. So SARS-CoV-2 is definitely a highly structured RNA throughout its length. So this Gorse analysis that one does is for a global analysis of the entire genome. And we decided to take it a little further and do this in a windowed way along the genome to see if we could actually begin inferring what were the most structured portions of the genome. And we are doing this because regions of intense RNA structure or high structured content are often regulatory in RNA viruses. And these are places that you might target with drugs or understand better the mechanism of the virus. So we used a partition function calculation in superfold, which is described in this paper, in order to do a very rough computational analysis of base pair content at discrete regions along the genome. And we correlated that with a calculation you can run called the Shannon entropy term. And that is the propensity for a region of RNA to form a single discrete structure or many discrete structures. And if you have low Shannon entropy, it's likely that you are making just one type of structure. Now, what we did is we plotted this high phi or high base pair content scores versus low Shannon entropy. And in regions where we have a high ratio of this, and I'll show you this on this heat map, numbers greater that are closer to 100% are more red. These are regions predicted to be highly, highly structured and have a lot of discrete secondary structure. And this is the whole genome, and I'm going to blow this up for you. And you can see here that in ORF1AB, there are many regions that are almost just completely static structure, have complete discrete static structures, especially in this NSP3 gene here. There are very few regions that are white, in other words, likely to be more like spaghetti. Almost the entire thing is very highly structured. And this is especially true in the part of the genome that encodes the non-structural, the structural proteins and the subgenomic RNAs. So the subgenomic RNA transcripts are even more stable and structured than the genome. So given this fact, we were often faced with this conundrum that the full genomic RNA is made as a single transcript, and it's floating around together with transcripts of the subgenomic RNAs, and in many places they share sequence. So I'm going to show you a cartoon of the sequence of the N that encodes the N protein. And you could imagine that the secondary structure of the RNA in here, when it's adjacent to this ORF8 RNA, is going to have a structure, a secondary structure that depends on its neighbor. And indeed, we see this. So we can calculate a hypothetical secondary structure of the upstream region of the N gene RNA. And we see that when it's next to ORF8, it has this structure. But in the subgenomic RNA, the upstream element has been cemented to a leader sequence, which has a completely different and autonomous secondary structure. And then the whole thing folds differently into a much more stable structure. So this is very exciting. It means that depending on the context, these SARS-2 RNAs can adopt completely different folds. And the cool thing is that the subgenomic RNA fold is much more stable than the genomic one, which may explain why subgenomic RNAs have higher stability, longer lifetimes, and it affects our design of COVID test kits, primers, and controls. I'm just checking my time here. Okay. All right, now all of this was very hypothetical. We wanted to actually map the secondary structure of SARS-CoV-2, especially the ORF1AB region, which would be unambiguous, in living cells. And until recently, this would have been impossible, but we had a couple of advantages in this particular study in which we intended to use chemical probing. One advantage we had in undertaking the study is that SARS-CoV-2 completely takes over an infected cell. It becomes about 70% of the total RNA that you isolate from that cell, which is pretty awful unless you're an RNA biophysicist and you want to study structure, because then you have a lot of RNA to work with. The second thing was that we had recently discovered and optimized a new reverse transcriptase. And this enzyme makes it possible to probe lesions on RNA chunks that are kilobases in length. So we can do chemical probing using amplicons that are thousands of nucleotides long. And we have other papers on this enzyme, and it's called Marathon RT. And we had just completed parallel work on the hepatitis C viral genome, so we had a technical foundation. And we are also part of an amazing local research community that we have collaborated with a lot of success. And I'll give you some pictures of them here. Our partners in doing this study are virologists Brett Lindenbach, Craig Willen, and their students. And these guys did the part of the work that I'll be showing you that takes place under BSL-3 conditions, because we have to probe the structure in infected cells, so we have to use biosafety level 3. Then once the RNA that we've probed is taken out of cells, lysed cells, and is cleaned up, it can come into our lab, and then it was analyzed by Nick Houston, Han Wan, and Shivali Patel, who are 3 extraordinary grad students in my lab. And much of then the downstream analysis was done by Rafael Tavares, who in phase 0 was at home, but the rest of us were in lab. So how did we do this? We used a chemical probing methodology where the chemical probe easily gets into cells and can modify the RNA. And what we're trying to do is just interrogate whether there are regions that form base pairs versus single strands. And chemical probes will react with the single-stranded regions, but not with the double-stranded regions. And the probe that you can use one of 2 probes, an acylation probe or dimethyl sulfate, and we're going to focus on this acylation reagent, which will modify any 2' hydroxyl group that's floppy. So in orange here, you see this nice RNA structure. A shape probe will attack these nucleotides, but not the purple ones, because they're involved in base pairs. How did we do this work? We took a SARS-CoV-2 isolate from a patient in Washington State. We infected Hu 7.5 cells with it. This is all in BSL-3. Through that to 4 days post infection. Then we treated with the acylation probe. This is the chemical probe that modifies single-stranded RNAs. And then through a reaction that I'll show you in a moment, we used the reverse transcriptase to identify the positions of all the modifications. And so we made amplicons using the reverse transcriptase, and we were able to cover the entire SARS-CoV-2 genome with only 16 amplicons, which is unprecedented. If you were going to do this with conventional sequencing technology, it would take hundreds of amplicons. And that's because conventional reverse transcriptases can only go a couple of hundred nucleotides before they fall off. But with Marathon, we could generate these robust 2,000 nucleotide amplicons. We do amplicon PCR, we would sequence them, and then we would use a downstream analysis I'll allude to. Now, we didn't look for whether or not the reverse transcriptase would stop at these lesions on the RNA. So what I've just told you is that in single-stranded regions, the chemical probe is going to put an acyl group on the RNA. We're using a technique developed by Kevin Weeks and reported here, in which the reverse transcriptase actually bypasses the lesion, but when it passes by, it drops a mutation. So it drops an inappropriate nucleotide at that position, and you can identify it by sequencing, by deep sequencing. So the whole thing becomes much faster. I'm going to go over this quickly because it's complicated, and if you want to do it, you would want to read this paper. But you basically take your reactivity input file, you calculate a base pairing partition function in Windows, you force probable base pairs, and then you plot the actual positions of the interactions. And then what we like to do is to plot the shape reactivity as a function of Shannon entropy in a plot much like this, and I'll get right to that. So here is the entire ORF1AB region, actually, yeah, of SARS-CoV-2. This is the first time, actually, at single nucleotide resolution, anybody has probed any kind of coronavirus genome and determined its secondary structure. And what you see here is, in vivo, I should say. What you see here are regions where the shape reactivity or the chemical probe reactivity is low, and the Shannon entropy is also low. So these bars here. And these are sweet spots for the likelihood of discrete structures. So the gray bars are what you want to look for if you want to find possible riboregulatory elements. So what we observe is an abundance of folded structure in vivo. It's consistent with what we saw with computation. And through methods that I don't have time to tell you about, we've found that these individual structures are conserved. And this serves as sort of an atlas of stable structures in the RNA genome for downstream biophysical characterization. And this diagram doesn't really look very satisfying, so I'm going to show you some real structures. So these are structures that we've calculated in the next few slides. I'm showing you the secondary structure of the 5' UTR and the 3' UTR. And you can see that in black here are regions where the shape reagent is telling us that it's perfectly base paired. And the colored regions are where you have the tips of loops. And what's nice is that this substructure for the 5' UTR is very similar to what had been predicted previously for studies of other coronaviruses. And the same thing is true for the 3' UTR. Our data support what's known generally for other types of beta coronaviruses. So those were, I mean, this is the first in vivo prediction actually for any coronavirus, but at least it's in line with what had been seen before for in vitro studies. Now here's some really new things. These are secondary structures that we have identified, or we've identified hundreds of them, but I'm going to show you a few. In region 23, which is in the gene encoding non-structural protein 6, you see this beautiful substructure that's highly conserved. Region 25 encodes this structure that I like to call the airport, and that we are probing now. And we find that when we disrupt this region with locked nucleic acids, we disrupt viral replication. Region 34 and 36 are also highly conserved. And one of the reasons we know they're important is that in regions where the structure matters, you're not going to see high levels of synonymous mutations in the double-stranded regions. So if you take, for example, region 34, the nucleotides in the loops are free to mutate, but the nucleotides in the double strands don't mutate, which indicates that, that they are conserved. And there's other metrics of conservation. And finally, the much discussed programmed ribosomal frame shifting element. That's an element within ORF1AB that controls the expression of that through programmed frame shifting by the ribosome. And in the past, the PRF has always been modeled to look like this. And using our analysis, you can actually determine if that's the most prevalent structure. And we find it's actually a minor conformer. And the major structure in that region looks something like this, where you have a long-range kissing interaction. And in fact, this region here forms base pairs with nucleotides about 400 bases away. So it's only a tiny fraction of the time that the virus is able to form this pseudoknot that induces frame shifting. So that has important implications for mechanism. So I'm going to wrap up and reiterate what we've learned and what we're going to do next. So the work has established that the most highly structured RNA genome ever studied is SARS-CoV-2. It suggests a high level of regulation at the RNA level, but we are now in the process of doing functional validation of the structures we've found. Once a replicon system is in place, and we're helping Brett Lindenbach with that, we will be able to use genetics to test these, but in the short term, we are using locked nucleic acids to disrupt these structures in infected cells. Also, I want to emphasize that secondary structure is only part of the story. Some of these elements are likely to form tertiary structures, and there are new ways that we're doing chemical probing and cryo-EM to look at those. There are also important sites of host and viral protein binding, and we'll be using CLIP and other methods to look at that. And any validated RNA regulatory element is a wonderful potential drug target, because now we know we can target RNAs with small molecules. So very quickly, I want to thank my students. I already mentioned them before. I also want to give a shout out to Akiko Iwasaki and Nathan Grubau at Yale, who've been very helpful. And I especially want to thank HHMI and the National Institute for Human Genome Research, because these guys funded the development of the reverse transcriptase that made this work possible. And thanks for listening. I welcome any comments or questions.
Anna, thank you for that beautiful talk. Can you hear me okay?
Yeah, thank you.
I have one quick question, then open it up to others. In one iteration of the SARS-CoV-2 RT-PCR testing, N1 and N2 primers are used. So they're, as you know, adjacent, non-overlapping. And in some instances, one primer works and another primer doesn't. And I'm wondering if one of the explanations for that variability could be the secondary structure changes.
We think, yeah, thanks for that question. We think this is a really big issue. The primers were designed before people knew what the secondary structure was. As soon as we got this data, we posted it on bioRxiv in like July. So it's been available, and I would urge anybody involved in development of the testing primers to look at it carefully, because I think it explains a lot. You know, I think most of them were determined empirically, but I think now it's time to take a good look, because many of these structures are very stable. It's going to be hard for primers to bind.
Hi.
You're muted, Andy.
Sorry, we have a question from Miriam.
Hi, I just wanted to know if you have looked how specific these structural elements are to the virus mRNA, and how similar or different they are from the human mRNAs, specifically because they seem to use human translation machinery. So I just wanted to know if you have looked at that. Thank you.
Well, we don't share much sequence at all with this viral genome, so we would not have been amplifying any human RNA. And again, the virus makes so much RNA, it dwarfs anything else in the cell. It's even more, it's on par, actually greater than our own ribosomal RNAs. So we don't know, though, how well ribosomes interact with this RNA versus human RNAs. But remember, our own messenger RNA populations are very rare. We don't have a lot of active mRNA that you can detect in the cell. So you know, the cell is basically, and its equipment is basically no match for this virus once it's infected. It just takes everything and uses it.
There's a question from Tom Gallagher. Do you have insights on RNA elements specifying gRNA encapsulation into virus particles?
Not yet. That's really important. That's a big goal that we have to, you know, we've only succeeded at this point in indicating which parts of the genome might encode things important for function. In the case of Hep C, we've been able to parse that into individual kinds of functions, but we're not there yet. But soon, I'm hoping, with better assays and replicon systems, we're going to be able to get at things like packaging signals. So that would be the next step.
Okay, Anna, thank you. If there are no more questions, can you unshare? And our next speaker.
I think Rodney has a question.
Oh, okay, sorry, Rodney.
And I may ask a question too if I don't overlap with Rodney.
Let's see if I can unmute Rodney. Okay, Rodney.
Hi, great. Anna, thank you for a wonderful talk. I wondered, we heard a little bit about some deletions that occur occasionally in SARS isolates, and especially in SARS-1. with the NSP8 gene, excuse me, the ORF8 gene. Have you looked at any of those deletions and asked how they change some of the structures that you predict, et cetera?
Yeah, thanks for that question. Thanks to David Hausler and his team, we've just uploaded all of this onto the UCSC genome browser, and it's correlated with SNPs, but I haven't correlated it yet with the deletions. We've just started doing these correlations. One of the things that's cool is that the SNPs that we see in the virus and mutational changes we see are almost only in single-stranded regions. We don't see much change in sequence that persists anyway, that occurs in any of these structures. So that's the short answer, but I haven't looked at the deletions, and I will do that. Thank you.
Eric, did you have a question?
Yeah. Hi, Anna. Thank you for giving the talk. It was great. Maybe this is a naive question or just irrelevant. I'm not sure. If you take your computational predictions for the RNA structure, can you expand on that and start looking across different coronavirus family members and ask whether there are discrete changes in structural units associated, for example, with distance, genetic distance or relationship? And would those be indicative of changes in regulatory functions?
Yeah, that's a very interesting idea. And the thing I really love about this computational pipeline that my student Rafael Tavares has derived is it's very fast, and we can easily do what you've suggested. You know, I think it's important to stress that the actual empirical measurements, the chemical probing, is expensive and difficult. Not everybody can do it, and you can't do it on all the viruses. So these computational methods are critical, because we could definitely do what you suggest and then help pinpoint what's really different about SARS-CoV-2 compared to other coronaviruses or beta coronaviruses, and to look at some, you know, kind of almost look at phylogenetic variation in structure as opposed to just sequence. So that's a great idea.